I'm Madison Evans.
And I'm Megan Evans.
And welcome to Crash Course Biology. Today we are going to talk about cell division.
Miss Evans, do you mean we're going to divide cells by cells?
No, me from the past. We don't mean that kind of division.
We're talking about the different ways that cells divide into two or more cells from one cell.
The method that a cell uses depends on the type of cell. For example, prokaryotic cells use binary fission as one of their ways to reproduce. In binary fission, the DNA is replicated and the cells begin to elongate. Eventually, the replicated DNA separates into two genetically identical daughter cells are formed.
Eukaryotic cells use different methods depending on the cell type. Somatic cells use mitosis, and cells that are used for sexual reproduction use meiosis.
Madison and I are going to go into detail about these two processes in this video. Before we can talk about mitosis, we have to discuss the cell cycle. The cycle shows the different stages of a cell. For the majority of the cycle, the cell is in the different phases of interphase. These stages are the G1, S, and G2 stage. In G1, the cell grows and functions normally until it reaches a certain size, and then the cell transitions into the S phase. In the S phase, the DNA is duplicated. The DNA is duplicated because mitosis requires two sets of DNA. In G2, the cell continues to grow and prepares for M phase, which includes mitosis and cytokinesis. There is also a stage called the G0 stage, in which the cell does not go and does not divide. Nerve cells and liver cells are some examples of cells that are used in this stage.
Let's go to the thought bubble to learn more about mitosis. The first step of mitosis is prophase. In this phase, the chromatids condense and the nuclear membrane breaks down and disappears. The mitotic spindle and centrosomes form to be used to separate the sister chromatids.
The next step is metaphase. In this phase, the chromatids line up in the center of the cell or at the metaphase plate. After metaphase, anaphase follows. In anaphase, the sister chromatids separate and move towards the poles.
The final step is telophase, where the spindle disappears and the chromosomes unwind. A new nuclear membrane forms around each group of chromosomes. The final part of this process is cytokinesis, which is not part of mitosis. Cytokinesis is the splitting of the membrane by creating a cleavage furrow in animals or a cell plate in plants.
Thanks, Thought Bubble. So the end product of mitosis is two daughter cells that are genetically identical. Multicellular organisms use mitosis to grow and to repair damaged cells, while unicellular organisms use mitosis to reproduce.
How does a cell know when to divide or not to divide? Sorry for the Shakespeare reference. The cell uses cell signaling to regulate the need to divide. Also, the cells must pass through several checkpoints.
We will start by discussing those checkpoints. The checkpoints are regulated by proteins in the cytosol of the cell. The first checkpoint is a G1 checkpoint. At this checkpoint, the cell is checked to see if it is okay to proceed to cell division. It is checking for the size of the cell, the DNA integrity, the growth factors, and if the cell lacks one of these things, then it will not divide and will enter G0 phase and will stay there until it can pass the checkpoint.
The next checkpoint is the G2 checkpoint. This is the final checkpoint before mitosis. At this checkpoint, the cell makes sure that it is the right size to divide and that the DNA was replicated correctly in the S phase.
Cells also use regulatory methods to control the cell cycle. In order for cells to grow, it must receive signals from growth factors. A growth factor is a hormone or a protein that promotes growth. Without the growth factor, the cell will not grow.
The final two regulation methods are the density-dependent inhibition and the anchorage dependence. The density-dependent inhibition regulation is when cells stop dividing once they touch each other. The anchorage dependence regulation is that cells must be attached properly to the surface before they can divide.
When the checkpoints and regulations methods do not work correctly, cell division can occur without regulation. This causes cells to irregularly divide, creating cancer cells and tumors.
Now it's time to move on to meiosis.
Oh, I think I've seen a square dance about this.
Yes, me from the past, you are correct.
Meiosis uses the same steps as mitosis, but it goes through the process twice without duplicating the DNA twice.
Let's go to the thought bubble to get a better picture of this process.
Just like in mitosis, the DNA is replicated in an interphase, and then the cell starts meiosis 1. Meiosis 1 is just like mitosis, except for a couple small details. In prophase 1, the homologous chromosomes line up together and undergo crossing over. This is when DNA from one of the chromosomes is switched with the DNA from the other. Homologous chromosomes are chromosomes that contain the same type of genes, such as chromosomes that carry different alleles for eye color. In metaphase 1, the homologous chromosomes line up across from each other. In anaphase 1, the homologous chromosomes are separated. Telophase 1 happens the same in mitosis and meiosis. Meiosis 2 happens right after meiosis 1 with no DNA replication. The steps in meiosis 2 are just like mitosis. Thanks, Thought Bubble. The final product of meiosis is 4 cells that are genetically different from the original cell. These cells are haploid, meaning they have half the number of chromosomes that a diploid cell has. These cells are later used in fertilization. Organisms go through meiosis to increase genetic variability through random assortment and crossing over.
Just like with mitosis, there can be problems with this process. It is called non-disjunction. This can occur in meiosis 1 or meiosis 2. If this occurs in meiosis 1, one pair of homologous chromosomes will fail to separate, and none of the cells will be normal. 2 cells will have 1 extra chromosome, and the other 2 cells will have 1 less chromosome.
If non-disjunction occurs in meiosis 2, then one of the pair of sister chromatids will fail to separate, and only 2 cells will be abnormal. 2 cells will have the correct haploid number, one of the other cells will have an extra chromosome, and the other cell will have 1 less chromosome. That's all the information we have for you today.
If you did not understand everything, feel free to watch certain segments again.
Thank you to our production team for making this happen. Also, please leave suggestions and questions in the comment section below. Thanks for watching.
So the end product of mitosis is 2 daughter cells that are genetically identical. Oh. Just like with mitosis, there can be problems with this process. It is called non-disjunction. This can occur in meiosis 1 or 2. If it occurs in meiosis 1, one pair of homologous